Hey everybody, welcome to the Lab Insider. I'm your host, Andrew MT. Our first video is going to talk about the history of medical laboratory science. We'll go through the step-by-step on what exactly it is that the science entails in the whole spectrum of medicine, as well as what the medical laboratory scientist or technologist actually does. Stick around. Alright everybody, welcome to the first show of the Lab Insider. I'm Andrew MT. Let me discuss a little bit about myself before I get into the whole logistics of the show and what the episodes are mostly going to be about. So, I'm a medical laboratory scientist. I work in a transfusion science laboratory. I'm specialized in transfusion science and immunological studies. A lot of you might think that's complicated, but it's really, when we break it down, just another discipline in medical science. See, there's many disciplines, many facets to medical laboratory science that not a lot of us know about, to be honest. There's, of course, transfusion science. There's clinical biochemistry, clinical chemistry, the study of molecules in your blood. There's hematology and hemostasis, which studies the actual components of your blood and its clotting mechanisms. There's microbiology, studying organisms, ooh, crazy bacteria and such. And then there's a bunch of specialties that a medical laboratory technologist can learn. Now, the reason I'm doing this type of a video, or this type of a series, is actually because not many people know what exactly medical laboratory technologists do and who exactly we are and how exactly we affect and how exactly we help the medical science community, or the medical community in general, I should say. We play a pretty vital role, but because we work behind the scenes, we're not so well known throughout the medical community and throughout lay people communities, basically throughout society. Like, everybody knows about your nurses, everybody knows about your doctors. They get the most respect, right? You watch shows like ER, you watch shows like Grey's Anatomy, and you always see the doctors, you see the nurses, you see the medical staff, you see the orderlies. They're all part of the whole hospital drama. But what you don't see is the lab techs. What exactly are the lab techs doing? Or are they even there? Are they analyzing blood? Are the doctors running blood tests on the samples that they receive, or the samples that they themselves draw, which, I'll be honest with you, it's the lab techs who draw them? Well, sadly, we don't get enough credit. So, I'm hoping that this series is going to bring credit to lab technologists. So as I sit here, I'm going to give you a little bit of a rundown of the whole idea of medical sciences, the whole idea of the study of biology, human physiology, basically, because it plays a pretty vital role. So essentially, what do we do as medical lab technologists or scientists? Well, we work in medical labs, so we work in hospitals, we can work in private labs and research labs. We use scientific techniques and scientific experimentation to provide analyses and results on patient samples. Patient samples being blood samples, body fluids, tissues. Um, can I think of anything else? Uh, Bone aspirates. And basically, we use these samples to help clinicians get a better picture of what the diagnosis of the patient may or may not be in general. Because you see, doctors don't know exactly what's happening inside your system until they actually get to interpret the results to come up with a way of treating the condition per se. So, for example, uh, your grandfather's a type 2 diabetic. Well, how did they know? How did doctors or your family physician find out that your grandfather's a type 2 diabetic? Well, simple. They drew a blood sample and they ordered a fasting glucose test. Now, that glucose test goes to the lab where a laboratory technologist receives the sample, checks for the sample integrity to make sure that it is a good sample, a good quality specimen, and with that, they actually test it in the lab. So what we do is very vital to the whole entire aspect of patient care. We have to really ensure that our results are precise, that they're accurate, and that they're going to give the doctors a general idea of what's wrong with the patient. So for example, that glucose result comes out and it's a high fasting glucose, let's say a fasting glucose of 14 millimoles per liter. Now, that is indicative of a diabetic patient, a very severely diabetic patient, and that can help the doctor catch that diabetic episode before it even turns into something like ketoacidosis or before the patient slips into a diabetic coma. Now, these are stat results, and medical technologists provide the results on a quick time demand. So basically, the sample enters the lab, and like this, we got to test it because the patient's waiting on it, the doctors are waiting on it, life-saving treatment is waiting on it. For example, where I work in a transfusion service, you can imagine that people losing blood, it's quite a serious scenario. It actually is. And the reason being is because we don't have a lot of time before the patient, let's say, bleeds out and loses a ton of blood. But at the same time, we have to make sure that the doctors who are going to help infuse the blood are infusing the correct blood group or a product that actually matches the donor's blood group. So what I do is I test not only the patient's blood type, you may have heard of A, B, O, AB blood groups, I also test the donor and I perform a cross match. And a cross match actually enables me to match the patient's blood with the donor blood and mix it together and see if it's gonna react somehow. So very important work. Very well, I wouldn't say undervalued by the medical community, but undervalued by most patients who are not aware of what happens behind the lab or behind closed. We do have, what do we call it, procurement facilities, however, and in procurement facilities, it's rather interesting because that's where we actually collect the samples, and we take pride as medical technologists in actually collecting our samples because of the fact that we have to ensure they're at the utmost quality, that the sample integrity is actually kept. And what I mean by sample integrity is I mean that we have a sample, a blood sample, let's say, that we can run properly on the machines, on the analyzers, that we can test manually properly. And the importance of having a good sample is that the results that we get in the end are quality results. And little disclaimer, because now I just realized as I'm looking at it, I apologize for the webcam quality. I will try to get better quality recording equipment as the future progresses. Like I said, this is just an intro video, a little bit about myself, a little bit about the history and the community of medical laboratory scientists, and just like an all-around scope of what I'm going to be talking about. So yes, just bear with me and bear with the crappy fuzziness of my shitty camera, I guess I would say. So let's get into the history of medical laboratory science. Medical laboratory science probably began around the turn of the century, industrial revolution type thing, where we did have scientists, we did have early Victorian era microscopes, and those looked at first versions of cells and biological things at a more microscopic level, but people didn't really have an understanding of the complex nature of anatomy, especially the complex nature of human anatomy at the microscopic level, because we have macroanatomy, which is our anatomy, but we also have microanatomy, and microanatomy is tissue structures and organs and how they fit together, how they work physiologically in our bodies. Basic structure being the red blood cell. Let me angle that forward. So this, being a stress ball, is in the shape of a red blood cell. The red blood cell is probably the single most important component in the human body. It is what gives us life. It is what carries oxygen through its hemoglobin molecules, and it is what we as medical laboratory scientists are most interested in, or at least on a hematology level, which means the study of blood. Basically, the red blood cell is at the source of much of what happens in our body. It's found not only in our vessels, but you can find it in sinuses and cavities within our tissues, oxygenating our other cells. So this is what actually sparked the interest in a lot of early scientists in discovering bloodborne diseases, because in early 19th century, I would say, the most common associated thought was that when you were sick, something was wrong with your blood. We didn't know what blood cells were until they were actually microscopically identified. But something was essentially wrong with your blood, and they had practices like bloodletting and a whole bunch of other weird stuff. However, it's not the oldest form of medical science, it's the study of blood. Actually, the study of urine dates back way further to like medieval times, I would say, or Renaissance times at least. So, urinalysis, what we call it, or urine analysis, is essentially the study of your piss, to put it bluntly. And the way old scientists and botanists and biologists, the way they would study it is they would smell it. So urine in healthy people versus urine in sick people has a distinct odor. They would taste it. Yes, I said taste it. Tasting urine basically to see if it's sweet. So if there's diabetes, like high level of glucose in the urine, which means your kidneys aren't filtering properly or they're not working properly, they're letting out large amounts of sugars. Um, See if it's yeasty. If people have yeast infections, it's one way of knowing is odor and taste. Color is very important. Color is actually something that in current day urinalysis we do still use. We assess the color of the urine and we basically have an idea of what's going on. Clear straw colored urine is essentially healthy urine, but like darker amber urine would indicate other things like bilirubin. Foamy urine would indicate like proteins, an increase in proteins, which shouldn't be going through. Red urine, murky urine, hemoglobin or blood, basically in your urine, and possibly bacterial infection. Blue urine, green urine, and you're all thinking, what freak is that? Blue and green urine, but it's super important, and it all dates back to very, very old methods of testing, and that is how medical technology was being built upon and built upon. These are all building blocks, essentially. Uh, One that's really relevant to my field of medical science, basically transfusion science, is Karl Landsteiner and the discovery of the blood group system. Now, Karl Landsteiner was a pioneer in hematology and immunohematology. That's what we call transfusion medicine. We call it immunohematology. Basically, Karl Landsteiner discovered that we have blood groups, and blood groups are basically protein molecules. They're called antigens, and antigens are on the surface of the red blood cell membrane. So these antigens, basically, Karl Landsteiner discovered, show up around, and they stimulate immune responses between other cells to produce what we know as antibodies that will attack foreign invader molecules and cause transfusion reactions, possibly death if it's a major discrepancy. And basically, Karl Landsteiner created Landsteiner's law, which dictates that people of one blood group may only receive the same exact blood group, and at the same time, if people have that antigen, so let's say people are group A, they have the A antigen, Karl Landsteiner said that they will indefinitely, they will indefinitely produce an anti-B antibody against group B red blood cells, which is why we can't give group B to a group A person and whatnot. And I'm getting into the logistics, and I'll probably have episodes about specific disciplines and specific methods of testing. This is I don't want to get too specific in this video, but this is just to show you how over the years this whole idea of medical science evolved, and we get to this level of understanding, especially in the I would say 1940s, where there's a whole revolution of the labs and of people working in the labs, and believe me, it's been a predominantly female op, operated field of work. I mean, females helped shape, not just males, but female scientists helped shape the medical laboratory science community as well. And they still play a huge, huge role as majority. I mean, most of the people I graduated with are all female laboratory technologists. So I would say it's mostly a female-dominated field. But so throughout history, there was no like in the 40s, when the men used to have better jobs than women or whatever. In terms of medical science, that wasn't true. Women contributed equally as hard as men did to the field and to discovery. I mean, obviously, you all know about Marie Curie discovering radiation and whatnot, but this is more based on our human biology and human anatomy. So it is a community with a rich history. And because I work in Canada, I'm part of the Canadian Society for Medical Laboratory Science. I have my pin here, CSMLS pin. Probably can't see it. So that's the Canadian Society of Medical Lab Science, and this year it celebrated its 80th year, so you can just tell how rich that history is. And I see old pictures. I'll try to post some up in the video. Old pictures, CSMOS, of Canadian medical scientists working in laboratories and what they looked like. And you know, a lab back then was much different than a lab the way it is today. They had their bench tops, their workspaces, but at the same time, they they smoked in the lab, they drank, and they ate in the lab. Obviously, these are against our current day universal precautions. This is a book of laboratory universal precautions, laboratory safety, written by the CSMLS. Other societies of medical science probably have it. The International Federation of Biomedical Science probably has it. Um, so people would eat and drink and smoke in the lab. Lab coats were slightly not as long. They wouldn't cover you as properly. Your personal protective equipment was probably not as good as it is today. Also, safety, people didn't tend to wear gloves because they didn't realize the risk of bacteria and whatnot, or contamination from viruses, illnesses, diseases. Um, at the same time, what I find interesting is that their work also involved unsafe practices that eventually became safer, like mouth pipetting. Now, what is mouth pipetting? Or what is pipetting in general? Pipetting is the act of aspirating a liquid with what's known as a pipette. And basically, once you've aspirated that liquid, uh, pushing that liquid out into a different vial when you want to mix specific volumes together. Pipetting is actually the most important thing a medical technologist has to learn how to do because it has to be precise, accurate. They have to dispense proper volumes. So the pipette is kind of like the lightsaber of the lab. That's what I like to call it for all you Star Wars fans out there. Um, so it mouth pipetting, because pipettes were made of glass, and some are still made of glass. They were very manual. Now we have automated pipetters, things that are a lot easier with piston operated handles. But back then, the only way to aspirate it was either through a bulb, which the invention of the pipetting aspiration bulb came out way later. But the only way to do it was to suck it up with your mouth. So say they have a vial of blood or urine and they have their pipette and they need to suck up 10 milliliters in that pipette, they would put it in their mouth and they would suck up the liquid, and then they would tap it with their finger at the exact volume, and they would release till it goes down to the volume they need. Now, say you aspirate too fast or you're not sure how fast the liquid's going to rise in the pipette, you could aspirate that into your mouth. There's no filter stopping it or blocking it at all. It'll go straight into your mouth and you'll be contaminated with whatever that sample is contaminated with: infectious disease, viruses, HIV, AIDS, whatever the case is. So thankfully, that's a standard of practice. That is no longer a standard of practice. If anyone does it today's day and age, we can lose our practicing licenses. Of course, I would never recommend that in any laboratory setting, whether it's in a third world country or first world country. It's just not a safe method of practice. So we're glad that it got rid of. But see, now my point is that this is how over the years the study has evolved. And the study has evolved with multiple different disciplines, which I will get into in a moment. I'm just gonna do a little video cut here and show you some images of what I talked about, and I'll get back to talking about the disciplines. All right, everybody, welcome to the last part of the video where I'm going to talk about the disciplines in lab technology. Now, when I talk about disciplines, I talk about the different specializations, the different types of specialty. Now, there's just a few main disciplines, and then those could be further broken down into subspecialties. So, a general MLT, or a general medical laboratory technologist slash medical lab scientist, will have to during their schooling years will have to learn these main disciplines, and will have to become familiar with the scientific techniques used in each of them. So these are, I might have stated them earlier, but I'll restate them. These are immunology and transfusion medicine. They are clinical biochemistry, hematology and hemostasis, microbiology. These are the main disciplines that are super important. And of course, I'm forgetting clinical histopathology. So when we start with transfusion medicine, what is it? Immunology. Immunology is the study of antibodies. It's the study of immunogenic reactions. It's the study of how white blood cells, particularly plasma cells, produce antibodies to form an immune response, an active immune response, and how that immune response works and how antibodies work in the body. Transfusion medicine stems from that because obviously when we have an antigen or a protein, an antibody is usually the counterpart molecule that binds to it. So transfusion medicine works in that way. So what the transfusion scientist does is, as I stated before in my example, cross matches blood, tests patients for antibodies to make sure that they have no abnormal, irregular antibodies that can cause transfusion reactions. It's a very important job, very important facet. It could be very time consuming because patients could have multiple antibodies and you'll have to go through a series of antibody identification panels to be able to identify what type of antibodies patient has. So that's transfusion medicine. That is all on the fundamental basis of Landsteiner's law, like I stated earlier. We then have microbiology. Microbiology is the study of microbes. So basically in microbiology, the microbiology scientist, microbiology technologist, and microbiologist will ultimately culture microbes. They'll culture them on agar plates, basically petri dishes with an agarose media, and will allow these plates to incubate and to grow and to kind of fester this bacteria so that when it grows into colonies, they can look at the colony morphology, they can look at it microscopically, they can perform biochemical testing on it, and they can actually determine basically what bacteria is infecting a person or causing disease in people. So microbiologists play a vital role in the detection of pathogens, and they play a vital role in the use of antibiotics and the non-misuse of antibiotics. Doctors don't just prescribe antibiotics because they feel like prescribing whatever antibiotics or they have a list. They go based on sensitivities, bacterial sensitivities to antibiotics, and the way that's determined is through laboratory testing, is through antibiotic sensitivity tests with bacterial cultures. So that's essentially what microbiologists do. They'll also contact the CDC, they'll contact Center for Disease, that's the CDC. They'll contact disease control centers in other nations and whatnot, and they will update the status on bacterial contamination, bacterial spread, pathogenesis of bacteria, and whatnot. So when you hear about plagues, like you hear about pandemics like the H1N1, Ebola, that's more viral, but Ebola, that's really these virologists, microbiologists that study this, and they grow it in a lab and they basically identified it, and that's how we have known cases versus non-known cases. We also have hematology and hemostasis. Two split sections, but like I said, hematology and hemostasis are kind of combined. Hematology being the study of blood, study of anemias, leukemias, blood-borne diseases, malarias, anything that has to do with, as I said before, the red blood cell membrane, the red blood cell itself, the white blood cells, the growth and the development, the maturity of red and white blood cells. So we study those hematology technologists study the morphology of the cells. They study how the cells are shaped. They study basically how the cells are physiologically working in the body. Are they working properly? Are they non-working properly? Then we have hemostasis, which is study of coagulation. So coagulation is basically blood clotting, your ability for your blood to clot. And when we say blood clotting, we essentially mean that you have proteins that are called clotting factors, which activate and they form a mesh-like kind of structure that's called fibrin, and that forms a solid base to plug up any cuts or bruises that you have. And that, along with platelets that release tissue factor, are all involved in what's called the coagulation cascade. So we have to study, do people have abnormal clotting times? Does it take relatively long for their blood to go from liquid to actually clotting? So that's actually something very important. Now um, we also have other studies, such as patients who take heparin medications or anticoagulant medications. It's important that we study the concentrations of these medications in their blood to make sure that they're not gonna, I would say, underclot. You know, for example, they're not gonna bleed out if they cut themselves. Um, which brings me to my other discipline, which is very similarly related in the sense that both labs are usually one lab, is clinical biochemistry. Clinical biochemistry is, as I may have stated earlier, the study of molecules in your blood. So that'd be macromolecules that make up important components that our body uses for energy, that our body stores, that our body uses for action potential, for synapses in our brain, for muscle movements. So these types of molecules, like for example, glucose, um, ATP, albumin, sodium, potassium chloride, the electrolytes. We do fats, so cholesterol, HDL, LDL, triglycerides, fatty acids versus non-fatty acids. We test for total serum proteins, but we also test for immunoglobulins, which are antibodies. So you could take that from immunology, and you can easily interchange immunology with biochemistry. Um, and biochemists can do urinalysis. Of course, we spoke about urinalysis before. Urinalysis has a big biochemical component where we study urea, creatinine, ammonia, all waste products that appear in the urine. Glomerular filtration rate is another big test for kidney function. We test liver function, so basically enzymes. We test ALT, AST, GGT, cardiac markers, which could also be muscular enzymes for people who've had heart attacks. Cardiac markers like troponin, creatine kinase. Um, so basically, that's the main scope of biochemistry, is the actual chemical part. And they use a variety of techniques. They use automated machines that do a lot of the testing through scientific reactions, chemical reactions, using a lot of reagents, which are reactive substances that are commercially prepared and bought or prepared in the lab. They perform chromatographies where they try to separate different types of molecules to be able to study them better. They perform electrophoresis. So that involves all of the chemistry type testing. Now, our last but not least is histopathology. Sorry, histopathology is actually the study of human tissues. So during surgery or an autopsy, for example, tissues are harvested, and basically they're put on a microscope slide. And the reason they're put on a microscope slide is so pathologists can analyze them and see what is wrong with the affected tissue. For example, a tumor is excised from the brain. It's then bisected and it's prepared onto a microscope slide, and basically it is stained, and then a pathologist will look and see where the cancerous cells are, what type of tumor it is, how it grows, is it malignant versus non-malignant. So histotechnologists, what they actually do is they get these tissues, they gross them, which means they look at them macroscopically, they analyze them, they take the dimensions of the tissue and whatnot. They look for abnormalities. They then segment them, they cut them or bread loaf them, and to very little segments that they'll then embed in paraffin wax. And then from that wax, they'll actually cut it very, very thinly, like 1 micrometer in length, which is nearly invisible to the naked eye, so that they can actually take that little um, piece of tissue and put it on a microscope slide and then stain it with a bunch of chemical stains so that we can visually see it, because cells on a microscope slide are typically transparent when they're cut that thin, so they need a stain for us to be able to visualize them. Colors in the human body are not very common. Blood has a distinct color because of hemoglobin, and there's some molecules that, there's some components of our body that actually do have um, their own colors, that do produce their own colors, but normally to look under a microscope we'd need to stain live tissues. So histotechnologists are responsible for providing very, very accurately stained, very accurately cut, very well preserved tissue so a pathologist can accurately diagnose the condition of the patient. So that is essentially all the main disciplines. The more specialized disciplines involve, of course, molecular biology, cellular genetics, cytogenetics, cytotechnology. It gets much more complex, and it branches out. But I just wanted to give you really an idea of how diverse a medical laboratory scientist is from one end of the spectrum to the other end of the spectrum. So with that being said, I'm going to end the first video there. I hope you enjoyed. I know it's a long video. I hope you stuck through it. I hope you subscribe if you want more videos. And yes, I will be posting more episodes of The Lab Insider. Thanks for stopping by, everybody.